Okay, we're ready to start the last session, which is optical imaging and contrast agents. For those of you who don't know me, I'm Jerry Glickson, and briefly let me tell you how I first met Jack Leigh. I was a postdoc at DuPont Central Research with Bill Phillips back around 1968, and we had the first superconducting NMR, and we were doing work on proteins. And one day, Mildred Cohn shows up, and Bill tells me I'm supposed to run experiments for her. And that was the day after my first child was born. I thought I had things under control, but I managed to screw everything up. So she was running experiments. She wanted to get the absolute maximum Boltzmann factor, so we were down just above 0 degrees centigrade. And I managed to forget to reload the dewars, so the whole experiment went kaput and so forth. A few months later, we're at a FASEB meeting, myself and Marty Poe, who was another postdoc there, and he had worked with Ron Estabrook here at the Johnson Foundation. And he's been telling me about this real sharp guy that got at JF, this guy Jack Leigh. And so we're having lunch with Jack, and he introduces me, and we start chatting. And Jack says, "Oh, you guys are from DuPont. You know, they have some real idiot running that 220 megahertz NMR spectrometer. He screwed up all of Mildred's stuff." And I introduced myself, I'm the idiot. So Jack didn't mince words. He told you exactly where you stood. So now you must have no confidence in what's going to be coming, but I assure you that the speakers today are of a much higher caliber. So now one other thing, I got to take some exception to this hairball analogy that Paul Bottomley has introduced. I just went through some bouts with hairballs on my favorite cat, and they only got two ways to go. They're either going to be spit up like John Detra just told you, or they're going to go in the opposite direction. So the thing with Jack was really much more sexually oriented. It's that he was able to produce offspring and launch people with careers that will have an impact for many, many generations to come. And that's really what we're showing you, is this seminal impact of Jack on the overall research community and of the numerous people that he either trained or he interacted with. Okay, without further ado, our first speaker is one of Jack's former MD, PhD students here, Perry Renshaw. Perry, for a long time, was the director of the Brain Imaging Center at MGH, and he's now moved to the University of Utah, where he can actually, instead of running a big center, can actually do some research. And he's going to be telling you about his early work on, I think, iron oxide, and also in work with lithium. Thank you.
Well, thank you all for staying. It's certainly a privilege to be asked to speak at such a significant day. My first confession is that I'm a coward, and so when I was told I should speak about contrast agents, I got frightened and asked Ravi for absolution, and he said I could talk about what I actually did, which is much more spectroscopy than contrast agents. So there's a little bit of bait and switch here. It is true that 2 months ago I joined June Taylor as the Salt Lake City base of former alumni of the Johnson Foundation. There are 2 of us now. Any of the rest of you who want to come out are more than welcome. Before I went to Salt Lake, I was for about 20 years in the Department of Psychiatry at Harvard Medical School. And I can tell you, based on my clinical training in psychiatry, that there are many, many more scary people than Jack Leigh. So I think he's been done a disservice by this particular symposium. Indeed, my recollection of Jack is that he was extraordinarily generous, because I have worked on only one problem for the last 20 years, and that is, one way or another, is spectroscopy good for anything if you want to do research in psychiatry? And dissimilar from Jack's other trainees, I was never very good technically. I tried building coils with Mitch's help, never worked. I tried running the scanner. I couldn't shim like Lizanne. I could, find blood cells for Jim Picard, but that was about the extent of it. So I had to do more applied work. And even in my later life, I've been fortunate to have many colleagues who've helped with some of the results I'll talk about today. So, spectroscopy in psychiatry, it is a niche area. I'm sure there are not 21,000 Google citations on spectroscopy in psychiatry. There may be 500 papers in this area. So it's not something you're going to have to hear about often or maybe even ever again. Psychiatrists usually study schizophrenia, so being an offspring of Jack Leigh, I study the other disorders, mood disorders and substance abuse. And if you sort of boil it all down, there are a few things you can study. Drugs, including lithium that I'll talk about, and energetics, because at least when I was here, understanding bioenergetics was important, and I'm embarrassed to admit that I still don't understand energetics, and so if any of you can straighten me out, after this session, that would be really good. Drug studies. I think it was Lotzi Julai, who was here this morning but had to go, who probably first suggested that lithium would be a good target for spectroscopy studies. It comes into play in psychiatry because we use it to treat people with bipolar disorder. Back in the day, it was reasonably easy to show that maybe people were grumpy in Philadelphia because they did not have lithium in Philadelphia city drinking water, at least as compared to many mineral waters, which actually do have reasonable concentrations of lithium. And in fact, it's a pretty simple experiment to get a resonance from the brain of someone who's taking lithium therapeutically. What can you learn if you do this? Well, once upon a time, I really was 75 kilograms, but that seems a long time ago. And you can figure out that it takes a while for lithium to get into either your muscle or your brain. If you take it therapeutically, and this box shows a relationship between a therapeutic serum lithium level and brain lithium levels on the y-axis, you can show about a threefold variation in how much of this drug is probably getting to the target organ in individuals with bipolar disorder, which is a bit disconcerting if you're a pharmacologist. We've begun to understand a little bit about why lithium does or doesn't get into the brain. One of the big factors that drives it is age. Your brain doesn't really like having lithium in it, and in fact, most bipolar patients will tell you they don't really want their mood stabilized, that it dulls them, it grays them, they don't care for it. And if you're a kid, it's going to be particularly true that you don't get lithium into the brain, which is sort of problematic because we in psychiatry are diagnosing more and more children as having bipolar illness earlier in life. Sure. You want me to switch to this, even if there's feedback? Better? No? Yes. How's that? All right. All right. Well, so much for talking fast and softly so that I get off the stage, I guess. Okay, so lithium in the brain doesn't get into the brain very well if you're younger. The brain's much better at pumping it out. Intriguingly, this relationship breaks down completely in old age, and probably we ought not to be using lithium very often to treat older adults. The other drugs you can see, of course, are fluorinated drugs, and this is my example of spectroscopy having an economic impact. Many of the drugs you see here we use clinically, with the exception of fenfluramine. These are all antidepressants that are fluorinated. We, of course, measure an area under the curve or a signal intensity and pretend that we can calculate a drug concentration by waving our hands really quickly. The technique is sufficiently expensive that there are relatively few validations of this technique. We did a study for a drug company that wanted their compound approved a few years back, and we were able to show that the green squares and the purple circles sort of aligned, suggesting that you could estimate a lot of those factors reasonably well. In this particular case, we were able to conclude that in overweight women who were taking dexfenfluramine at a dose of about 10 milligrams twice a day, they had brain concentrations of drug plus metabolites of 4 micromolar. And this was actually a significant observation because of studies done in a number of different animal species suggesting that if in fact you took dexfenfluramine or were administered dexfenfluramine, you would deplete your brain serotonin levels. And so you could actually deplete close to 80% of the serotonin in your brain with high enough doses of dexfenfluramine, whereas in humans, the range that we calculated with therapeutic usage was right up here. And on the basis of this, in part, a drug called dexvenfluramine or Redux was approved. It was developed by American Home Products just across the border in Delaware. Within a year, it was clear that the drug did cause primary pulmonary hypertension, and liability related to the sales of this drug caused American Home Products to go bankrupt and to be reborn as Wyeth Pharmaceuticals. So you really can have an impact with spectroscopy, but you can't guarantee that it's going to be necessarily a good thing. And we had nothing to do with trying to assess serotonergic function in the heart and the lungs. So from the context of running a big brain imaging center, these are nice studies to do. People come and pay you money and yell at you if you don't meet your timelines. I've gotten out of that business in part because the research that I'd like to focus on is really what goes on in people primarily who have mood disorders. And the heretical idea I have as a psychiatrist is that these, like many of the disorders that Jack and his followers have studied for a long period of time, are metabolic disorders. I don't need to introduce phosphorus spectroscopy. We've heard about it already today. We've even heard about the creatine kinase reaction. And Joanne, if you think creatine kinase is unusual and something as organized and regular as muscle, in the brain it's even more puzzling, at least to me. So here's a spectrum. And we started out with the idea that maybe there would be something wrong with energy utilization in the brain in patients who were depressed. And the reason we thought that was because about 20 years of blood flow studies had all suggested a downregulation of energy utilization in the brain. And so we did this study, and of course we got a result which must be wrong. And that was that ATP levels were decreased in the basal ganglia of untreated depressed patients by about 16%. Worse yet, here you have this depletion in beta NTP or ATP. There was absolutely no deficit in phosphocreatine. It was sitting there perfectly happy, and if anything, it was increased. So for a while I was banished from the spectroscopy community until there was a replication study the following year by a group in Germany looking at the frontal cortex, and they found exactly the same thing. That if you're depressed, and in this case in the frontal lobes, you end up having a deficit in ATP that's pretty significant. While there's really nothing wrong with your phosphocreatine, but in fact, if anything, it's probably elevated. So nothing much was done with this, and so we waited for a while, got a higher field scanner, and said, well, let's do it again. This time we looked at really depressed subjects, just to make sure that anything we might see would be meaningful and real. You probably can't see this, but here the depressed subjects in this column had elevated phosphocreatine and a tendency towards decreases in all of these nucleoside triphosphate resonances. Here the depressed patients, here the controls. A clue in this study was that the depressed patients also had markedly reduced free magnesium. So then we looked at people who got better and people who didn't. If you were going to get better with treatment, you would start off with a low NTP level that would go up dramatically with treatment, and you'd have a high phosphocreatine level that would decrease subsequently. And in fact, in this particular study, you had about a 90% chance of responding just based on an assessment of your ratio of high phosphocreatine to low NTP. It was as predictive as anything we've ever discovered in the area of psychiatry. Not that we can understand it. So if that's true, you want to do one of two things. One is you'd like to do what these blue lines are doing, that is the people who are getting better and increase ATP levels and decrease phosphocreatine levels. Or, as is so often the case with spectroscopy studies, maybe you want to do the opposite. Maybe you want to give people high phosphocreatine levels and low ATP levels to start with so that they can get better. And this is one of the problems I'd like to untangle. Our first attempt to mess things up started off with the idea that, well, we could give people ATP. And in Japan, ATP is included in many, many vending machine drinks. It has essentially no biological impact whatsoever, but it's very popular. Then we thought, well, maybe we could give adenosine, because in ACLS, you infuse it, and it makes people feel like they're dying, and then their hearts correct themselves sometimes for their rhythms, so that wasn't very good. But in psychiatry, we have for many years had the idea that a natural product, S-adenosylmethionine, that goes around and methylates things is an antidepressant. And in fact, there's some evidence that this is true. So here it was. We were going to give SAMI, and people's ATP levels were going to go up, and everything would be really good. Phosphorus spectra. And of course, as is so often the case, at least in my experiments, the opposite happened. that the effect of S-adenosylmethionine was to significantly increase phosphocreatine and to decrease ATP level in the brain. And there actually is a biochemical pathway that explains this. One of the things that SAMe methylates is a precursor to the synthesis of creatine in the brain, and so this sort of made sense. If this was true, and this is what you wanted to do, you wouldn't have to fuss and bother with SAMe. You could use creatine, which is an over-the-counter supplement that's widely marketed in the United States for exactly the same purpose. And this has been studied in muscle to a large extent and in brain to a lesser extent. You can see it both with proton spectroscopy and with phosphorus spectroscopy. Shown here, phosphorus proton spectroscopy. And what you find is that if you give healthy volunteers creatine, creatine goes up in proton spectra, shown here, and stays the same in patients treated with placebo. When you get creatine, you get an increase in phosphocreatine, just like with S-adenosylmethionine. You get a decrease in ATP. And so creatine does, in normal volunteers in the brain, exactly what S-adenosylmethionine means. Now, is this a good thing or a bad thing? That's what we'd like to figure out. There is one paper, a terrible paper, in which there were 11 subjects with a range of different diagnoses who were depressed when they started and were given creatine over 4 weeks. And you can see that some got better and some didn't. There was no placebo arm, so we can't really tell what was happening there. So I think it's an open question. We are doing studies both in animals and in man now. If you give creatine to male rats, it is profoundly depressogenic. They get much worse, they get much more sluggish, they get much more lethargic. If you give it to female rats, it's profoundly antidepressant. They get less anxious, they explore more, they're very happy, they're all over the place. So we don't really know. We're doing a clinical trial now with a collaborator in South Korea, and maybe in a year's time we'll know more. But ideally, you'd like to show that knowing a little bit about bioenergetics, or perhaps more to the point, what might be wrong with patients based on spectroscopy findings, would lead to something that you could do that would be helpful. The next steps we're taking are remarkably like all the things that were being done when I was in graduate school. And this is sort of how psychiatry works, slowly, and sometimes we go backwards. So what we'd like to know is if phosphocreatine and ATP are messed up, how else could we study this, perhaps in other organs or in other ways? So exercise studies that Jack did with his own homegrown device and that Joanne has done at Harvard for many years, we're implementing. And of course, we're trying to measure rate constants, forward rate constants for the creatine kinase reaction. So nothing's known. Is muscular function impaired? Is this a widespread mitochondrial defect? Is this systemic? If brain function is impaired, what's the relationship? That's not known. We can make measurements of the forward rate constant, at least in slices of the brain in this particular instance, using saturation transfer and watching the alpha resonance regrow. We're hard at work at this. Our preliminary results suggest that, number one, the older you get, the slower your creatine kinase reaction gets in human brain. And if you happen to have a mood disorder, it gets slower yet. Whether this makes sense to you, I couldn't say, but that's what we're finding. So, after 21 years as a grown-up, having left the laboratory here in 1988, I'm finally doing experiments that I could have done in graduate school if I was smart enough. And I'd like to blame Jack Leigh for not suggesting the right experiments to me, but that would be incredibly uncharitable. I have a little bit of time. There is another mood disorder we study, and that is the affective disorder of learned societies. Bipolar disorder runs in high-functioning families and amongst really promising and talented individuals, and can be a pretty awful disorder to have. We've used proton spectroscopy to study it, and unfortunately, all the things we measured back in the day, these peaks here, are not nearly as interesting as peaks that are buried in the noise, creating certain problems. Our model of bipolar disorder is that mitochondrial dysfunction plays a big role, at least in explaining a whole host of findings reported in the literature using proton and phosphorus spectroscopy. Fortunately, there's some parallel evidence from gene expression studies suggesting that if you have brain tissue from individuals who've had bipolar disorder, all of these boxes in blue show profound downregulation of gene expression in proteins that are involved in oxidative phosphorylation. And it always helps to have some basic science to support a spectroscopy grant. So we did a study and found some unmedicated bipolar patients. We used an echoplanar spectroscopic imaging sequence. And what we found was an increase in glutamate and glutamine in gray matter, but not in white matter, and an increase in lactate, again in gray matter, but not in white matter, in untreated bipolar depressed individuals. If this was real, would it go away with treatment? Well, maybe. There are standard treatments for bipolar disorder, lithium and valproic acid. Lithium tends to reduce glutamate and glutamine. Valproic acid has a weak tendency to reduce lactate in the brain. So that was sort of interesting. And if this was true, again, the leap of faith was, if this is what's wrong and this is how medications fix it, what else could we give that might fix it? We settled on some simple natural products, pyrimidines, both cytidine and uridine, in part because of an urban legend that these natural products actually are useful in treating mitochondrial dysfunction. They also do some interesting other things, that they increase the synthesis of neurotransmitters in the brain, and they surely speed up the rate at which phospholipids are synthesized. If we've learned one thing, and I've learned one thing in doing human studies, it's that they take an awful long time to get anywhere. So one of the principles that we have embraced wholeheartedly is that we need to really do translational studies. My former collaborator, Bill Carlazon at McLean, swore he could assign these emotions to these animals, but I remain skeptical. More concretely, Bill was an expert in something called the forced swim test. This is the test with the single greatest success rate for predicting antidepressant efficacy. What is it, you might ask? Take a rat on a nice day, put it in a warm plexiglass container, let it swim for 15 minutes. Take it away, put it back in its cage, dry it off, bring it back 24 hours later. Let it take another swim. Carefully study the behavior of the animal during a 5-minute swim. The extent to which it swims, climbs, and stays still has almost 100% success rate in predicting compounds that will be antidepressant. It does have some false positives. Things like cocaine and amphetamine will test positive as antidepressants, but this is widely used in the pharmaceutical industry. So cytidine, shown here, actually behaves a whole lot like a tricyclic antidepressant. So we thought we were onto something. An agent that has pro-mitochondrial effects and pro-neurotransmitter effects tests positive in an animal model. This is probably not an effect that's going to be restricted just to cytidine. There are a whole bunch of these pyrimidines, some of which you can find in health food stores and some not. And the reason why there's probably shared effects is because cytidine by mouth, by the time it crosses the gastric mucosa, has been turned into uridine, which is the form in which it gets into the brain, and it can be reconverted back into cytidine in the central nervous system. Trying to get any sensible clinical trial, especially one that's interesting, approved by the local IRBs in Boston, is hard. Fortunately, I have a colleague who went back to Seoul National University for family reasons, and so we were able to do a clinical trial there. And what we find is, in fact, if you give a bipolar depressed patient cytidine versus placebo, they get better about a week faster. Big effect? Well, it's not this. You're not getting better right away. But it is in the right direction. There are no side effects to this. It's going to be cheap as anything as it becomes approved on the GRAS list. And if you, in fact, are depressed, you probably would want to feel better a week earlier, other things being equal. One of the other effects that it has that we thought was quite interesting is that if you take cytidine as opposed to placebo, you get a tremendous reduction in levels of glutamate and glutamine. And that's shown with the lighter blue squares here. That neurochemically, whatever else this natural product is doing is it's having profound effects on this weak organic acid that plays a big role in excitatory neurotransmission. And that the effect is even larger in those patients who get cytidine who are going to have a positive response to treatment. That is almost certainly more than you ever wanted to know about the use of spectroscopy as a psychiatrist. I thank you for your kind attention in the afternoon. And we'll turn it over to the next speaker, who will surely talk about what he is supposed to talk about rather than what he wants to talk about. Thank you.
We'll give that to Bruce. Our next speaker is Bruce Tromberg. He is professor of biomedical engineering at the College of Medicine at the University of California in Irvine, and he's also the director of the Beckman Laser Institute at that institution. Bruce will introduce himself, no doubt, but he's one of the really top people in the optical imaging area. And Britton Chance and Jack Leigh started it here, and he has really moved the field ahead. And he runs a research center and sits on the board of Jack Leigh's research center. So he's had a very great deal of interaction with Jack Leigh during his career.
Thanks, Jerry. Okay, thank you very much, Jerry. Thanks, Mitch. Thank you, Susan. Thanks for inviting me. Just to give a little bit more background on my interactions, I'm kind of an outsider in a sense. I'm a little bit of a different point of view compared to the rest of the speakers. So my interactions with Jack really began in the early 90s when I came up to Britt's lab to give a seminar. At the time, photon migration or photon diffusion was this exciting new thing that really we were all trying to understand together. Arjun was a newly hired assistant professor. I can't remember, John, were you here in 91 or? Yeah, so John was a med student. And it was a very exciting time, and there was a lot of kind of shared interest and bonding that took place just in a day. And so we've maintained that relationship over time. In the mid-90s, I became an RR director, which is a kind of shared pain as well that Jack and I had together. Jack bore that in a very unique way. So we would often go to these PI meetings, and I'd find Jack in the hallway, not in the meeting. Of course, I guess I wasn't in the meeting either, but thinking about what Lizanne said, it was really interesting. He somehow would figure out what you were interested in and what you were good at, and that's how he would talk to you. So I started talking to him about the Phillies and the Eagles, and he seemed to be really into sports. And I asked Susan if that's the case, and she said, yeah, he talked sports to me, but I don't know if he spoke sports with all of you. It seemed that was what I was interested in. We talked about that at the RR meetings, really never anything else. So that was another introduction of mine to Jack. And we had a lot of fun doing that. I became a member of the External Advisory Board, which is an interesting position when you're in Jack's advisory board, because of course Jack doesn't like to go to those meetings. He likes to kind of pace out in the hallway. So it's a relatively easy job if you can get here and get the meeting together. So those were interesting interactions. And I think another thing that, as I reflect on this, another reason why Jack liked to talk to me is we shared a kind of a common, this occurred at Britt's 90th birthday. And I gave a talk, and I kind of teased Britt a lot. And I think Jack really liked that. I mean, he talked to me a lot after that, and he really just kind of appreciated the humor and the good-natured teasing of Britt. And I get the sense that he has done that for many, many years. So that was a common shared interest of ours. So as I think of Jack and I look at his publications and I think of my personal interactions with him, really kind of brains and brawn emerge. And so that's the nature of the title of my talk. I will not speak about contrast agents. I will stay in the spirit of Jack and talk about whatever the heck I want to talk about. So Susan sent me this picture. Oh. Susan sent me this picture, and I really like this picture because to me it sort of personifies this brains and brawn type of characterization. So there he is, smart guy, fit, it looks like he can take on the world. So I really, really like that picture. And of course, coming from California, there is another character that sometimes comes to mind as, having brains and brawn. But, in fact, he has nothing in between, so no comparison to Jack. So sometimes Jack, of course, was caught in between brains and brawn. It wasn't always smooth sailing, and I really am amazed at how he managed to navigate a variety of treacherous waters. And I think in his research, he also managed to bring in extremes, these notions of strength and intelligence. And in some ways, his interest in nuclear as well as electronic movement captures that diversity, that promiscuity of his intellect. He was interested in all spectroscopies. I'm not really clever enough to be interested in all spectroscopy. I only care about outer shell electron transitions. I was talking about this to John before. I really don't know or care that much about all of these other things, but Jack seemed to know all of the spectroscopies. Not only that, he wanted to apply them to every body part, although brains and muscle tend to emerge as the primary theme. So, these are kind of the two opposing extremes, the juxtaposition of MRI with beautiful images and optics with multiple light scattering and crazy tortuous photons, which John will talk about. And, of course, with optics, there's a great simplification. We don't really have to do high-resolution spectroscopy. We don't have to do high-resolution imaging, which for some people is great, and for others it's very frustrating. So what we are encountering is multiple light scattering. This is one of my students with 4 LEDs embedded in a spoon. These are, dollar LEDs or so that you can get in an electronics store. And the camera is a webcam. So you can see there's a tremendous amount of near-infrared light that's available that comes through centimeters of tissue that's available for detection, has reasonably high signal-to-noise, unlike some other technologies which will be unnamed here, that Jack was quite interested in. So there's a lot of potential, a lot of excitement, and you can see where we were back in the early 90s, thinking that, with some modulation approaches, temporal, spectral, spatial, perhaps we could squeeze out quantitative information content and do tomography. And John will tell you all about the tomography part. I'll focus a little on the spectroscopy today. So the major technologic advance really occurred, and this is why I went to Britt's lab in about 91, was his paper with Jack in PNAS on time-resolved in vivo hemoglobin spectroscopy. And essentially, this was the first attempt to use a pulse-based method, a temporally resolved method, to separate absorption from scattering or account for the incredibly long path length that photons take in multiply scattered media. It also integrates photon diffusion models, and they were really quite primitive at the time. So as I mentioned, this quantitatively separates absorption from scattering in principle, and it allows us to map, image, and do tomography of biochemical composition and structure. So let me just briefly introduce the techniques behind time and frequency resolved approaches and put them in the context of what we're working on. So in time resolved methods, one introduces a pulse of light into a turbid medium, into tissue. And we look at the temporal point spread function, the dispersion of that pulse in time. And the more dispersion, the more paths that are available for the photons to take, the longer it takes for the light to come out, the more scattering that medium happens to be. Now, we can also spatially resolve a measurement and again measure a point spread function. But here we just put light in one spot. And you can image that, or you can put multiple detectors on a surface. And that remitted light, if it's measured as a function of distance away from that source, again is diffusing as a consequence of these multiple light scattering events and some loss. So absorption lengths are very long, and that's the loss mechanism. Now, in the frequency domain, where Britt tends to like to reside, we know why Britt is there because it's cheap. It's really inexpensive. But I like it just because, I don't know, somehow I thought it was cool. What you do, and what we have been doing, is looking at some modulation transfer function as a function of frequency, temporal frequency. So we'll modulate light over many, many frequencies and look at the dispersion of these modulated waves. And these are scalar photon density waves. So they're not electromagnetic waves. They're carrying photons. It's kind of a carrier wave, but once they go into tissue, they actually bear information content. So we measure that dispersion, and of course, these two, the temporal and frequency domains, are related to each other by the Fourier transform pair. And the same thing can be said in terms of the spatial domain, but recently we've introduced an approach that measures this modulation transfer function as a function of spatial frequency or repeatancy in space. And here, again, we measure a dispersion, but we look at some spatially patterned light that's projected onto tissue. And I'll show you an example of that in a second. So the instrumentation that we and others have tried to develop, the goal is to try to get spectroscopic measurements from tissue. One would like to recover the full absorption spectrum quantitatively and the full scattering spectrum quantitatively from a tissue. And working backwards, what we've done is we've combined a broadband frequency domain technique where we modulate diode lasers over multiple frequencies up to gigahertz. And we combine that with a white light source, which is broadband in spectral domain, so that we can combine these approaches mathematically and recover the full absorption and scattering spectra. And that can be integrated into relatively simple instrumentation. In spatial frequency domain, we use digital light projectors or spatial light modulators to project patterns of light. We shift these patterns in phase and can demodulate the images. We image with a simple CCD camera. And because tissue is a low-pass filter, as we change the frequencies, there is different penetrance, and each frequency carries a little bit different information about the absorption and the scattering of the tissue. So this is a way of doing wide-field imaging and mapping of absorption and scattering, and possibly even tomographic imaging, because there is exquisite sensitivity to depth, because, again, tissue being a low-pass filter. So when you take these techniques and you make a measurement, what is it that you ultimately measure? We're going to get absorption and scattering spectra, separate them very nicely, hopefully have spatial resolution, and then ideally, we'd like to map hemoglobin, oxy and deoxy forms. Methemoglobin has a very beautiful signature. Of course, we can form indices, adding them together, looking at ratios in terms of saturation. We can measure water. There's a very beautiful near-infrared signature of water, and spectral shifts in that signature tell us about protein-bound water and deep tissue temperature. We can measure lipid at about 920. There's a very nice harmonic of the CH fundamental where we can pick up lipid information content. Cytochrome, tissue scatter power. This tells us about cells and extracellular matrix. And of course, we can look at exogenous probes. So, Jerry, that's my mention of contrast agents. Now, let me proceed with the vignettes that I'd like to share. And sticking with the theme of brains, brawn, and a few other things in between, I'll try to not go into a lot of detail. I've given you all background now because you're all experts in diffuse optical imaging. In a few minutes, you'll be experts in diffuse optical tomography from John, from his Chalk Talk. And I think I'll just move through a few vignettes on this fundamental theme, brains, muscles, and some other organs. Okay, so first of all, this is Britt, again, a promiscuous technology advocate. He's happy to put his head either in the path of picosecond pulsed photons or in a magnet with very little space for getting his head in. So he's okay with that. I personally would rather have, this very nice, simple interrogation of a light source. But what happens when you do this? What's the kind of information content that we can acquire? So here's an example. This is work from my colleagues at UC Irvine, Enrico Graton, Bill Mantulin, and Antonios Mikolas, who is at the company ISS, which makes a commercial device, a near-infrared oximeter. Looking at their overall goal with this is to look at brain function. And in particular, they're looking at two populations, normals and subjects with sleep apnea, obstructive sleep apnea, which is an area, if you look at Jack's CV, he was involved in that before anybody thought that you should be involved in that, something like the mid to early 1990s. So this is a very interesting and simple experiment. I think all of you might have to do this towards the end of my talk because of the therapeutic effect. If you hold your breath and you have these optical probes on your forehead, and you simply look at the changes in oxyhemoglobin and deoxyhemoglobin. So hold your breath as long as you can. Your brain does not want to be starved of oxygen. It needs oxygen. And it will require that you perfuse the cortex so that you can begin to be refreshed, you're not starved. You see a washout of deoxyhemoglobin because of this dilution effect. So this perfusion is the revival component that's necessary, autonomically instigated as a result of breath holding. And this is the type of response you see for a normal subject. This is simply the respiratory signal that shows you breath holding and now release. So you see this volunteer breathing. Now, this is an obstructive sleep apnea subject who has significantly altered hemodynamic responses to breath holding. So this patient appears to have severe vascular disease, and this can be detected in a relatively simple bedside type of manipulation. So with breath holding, and in fact this subject is not really able to hold their breath as long as the normal subject, you see that there is a decrease in oxyhemoglobin and an increase in the deoxyhemoglobin. So there is really an inability to provide new flow, new blood that's oxygenated to the brain, at least in this region of measurement. Which means that in order for the cells to continue to function, they have to extract oxygen. And you see the increase in deoxyhemoglobin at the expense of oxygenated hemoglobin. So eventually, after they release their breath hold, you start to see that there will be a normalization and a change, but the time constant for this is extremely long. So this is a very simple manipulation that can be done with relatively inexpensive bedside technology. So what is actually going on there? You know, from the MR community's point of view, those aren't that satisfying because we don't have any images at all, and there is not that much hope of getting an image out of a very simple experiment like that. Although I have recently seen a graduate of Penn of this program who worked with Arjun, Joe Culver's work in showing functional hemodynamics in visual cortex stimulation. I just saw his work over the summer at the Gordon Conference, and the images, the resolution of the images and the temporal response are spectacular. They rival what you see with fMRI, so I really recommend that you take a look at his work. But another way to look directly at what's happening during these vascular manipulations is to do imaging using cameras, that technique that I mentioned, the spatially modulated approach, through skull. So many of you are familiar with intrinsic signal optical imaging, which is done without any special modulation of light sources and detectors. But we can now quantitatively map absorption and scattering at multiple wavelengths by using these spatially modulated structured light projection patterns. So here's an example of an animal model where we're looking at the skull and measuring quantitatively deoxyhemoglobin, the sum, total hemoglobin, and saturation, baseline in our rat model, and following middle cerebral artery occlusion. So this is a stroke model. And really, quite dramatically, within 20-25 minutes, you see a tremendous drop in saturation. We see that the deoxyhemoglobin increases quite significantly because there's no more arterial supply. So in order for cells to remain viable, they have to extract oxygen. So you can see all of these parameters follow commensurately. It's a very, very dramatic way to visualize alterations that are ongoing when there are vascular changes in the brain. Another example is to look at cortical spreading depression. And this is a really fascinating experiment because the implications are we can measure scattering and absorption. From the absorption comes all the hemodynamic parameters. From the scattering, there's a lot of debate. Is that an electrophysiologic signal? Can we measure that on a time scale that's fast enough? Well, they're actually slow-moving electrophysiologic signals, of course, so one can capture those in cortical spreading depression, which are these slow-moving depolarizations moving across the brain. So I'll play you a movie now of a slow-moving cortical spreading depression across rat cortex. This work was done by my grad student, David Kuchia, who's he actually just started a startup company based on this technology. He liked it so much. That was his PhD project. So what I'll do is sort of point out these different panels. The first one is the scattering coefficient at 800 nanometers. And what you see is that there is a drop in scattering as this wave moves across the surface of the cortex. And what I'll try to do is freeze. OK. So this scattering depolarization wave is the leading edge, again, of that depolarization phenomenon. Now behind it should come metabolism, some cellular oxygen consumption. And in fact, that's what we see. We see an increase in deoxyhemoglobin coming right behind that wave. So this works because we can separate scattering from absorption. If we don't separate the two, we don't see that effect that's electrophysiologic. We combine that separation and we see the equivalent of the Bold effect and optical Bold effect, both in terms of deoxyhemoglobin and oxyhemoglobin. So this increase in deoxy occurs at the expense, the reduction of oxyhemoglobin. So it makes for a very, very interesting way and a nice story. And some of the challenges are to move this as fast as possible so one can look at functional activation in smaller and smaller regions. OK, so that's all for brain. And now on to brawn. So in some collaborative work with Doug Wallace, who's a pioneer in mitochondrial genetics and mitochondrial metabolism at UCI, and Jason Ruth, who is a former undergraduate and biomedical engineer in my lab for a year, and now graduate student here. He's sitting up here. He's decided to come to Penn because there's outstanding optics and photonics here, among other reasons. But this is work that Jason led during his time at UCI. And this was inspired again by Jack and Britt and work that they did in the early 1990s. One time in a fairly cryptic conversation I had with Britt, he told me that he had Greg LeMond in the lab. And I just was trying to figure out, why did you have Greg LeMond in the lab? And because I remember Greg LeMond is a cyclist. He retired from cycling. He won the Tour de France. And he said, well, he can't ride anymore. He has mitochondrial disease. That was the first time I really heard of mitochondrial disease. And I saw this work that they were trying to do, which is essentially to exercise Greg and others to failure. And the hypothesis is that with mitochondrial disease, there's an alteration, of course, if your mitochondria are unable to facilitate oxidative phosphorylation, then there is an alteration in this oxygen tension gradient from mitochondria into the vasculature. So if one is exercising or working or inducing a load on the muscle, then there'll be hyperoxygenation. So we kind of set out on an adventure to see if we could see hyperoxygenation in subjects with mitochondrial disease. And what Doug Wallace is interested in is the link between mitochondrial disease and diabetes. And could one develop these types of assays to assess individuals' risk for diabetes and mitochondrial disease? So I'll just give you, again, a few vignettes. At the time, the instrumentation that Britt and Jack were using was fairly primitive. Now we've advanced things to recover the entire spectra of the tissue, both the absorption and the scattering. And we have been able to look at muscle composition in different subjects. And it's quite interesting. We're beginning to see differences appear in different populations. So here's a normal subject. Here's a subject with mitochondrial disease and a subject with diabetes. So these are quantitative absorption and scattering spectra. We have about 20 patients now. We're seeing roughly the same trends, although it's just very early on in getting this data. But it's interesting that we can see the lipid peak appearing in this diabetic subject, where one might expect to see additional subcutaneous lipid, and perhaps what we're hoping to see is actually lipid representation in the muscle. So there's a significantly higher water peak. There's also changes in the oxy and the deoxy, much higher in the normal subject. And under exercise load, which is really what we're after, and this is how we've set up. Jason rigged this up entirely. There is a force transducer. Here's the optical probe. Here's the instrumentation to acquire the measurements. You can see the display of the force transducer. This is the kind of stuff that we're seeing. So one can squeeze the transducer to 70% of an individual's full capacity. And part of the challenge for this is to devise reproducible schemes for loading muscle. And I'm not sure, in those early days, there were a number of ways that muscle was challenged, not necessarily in a consistent way. So what we're trying to do is load muscle. We see deoxygenation of oxyhemoglobin and increase in deoxyhemoglobin during the loading process. And what are we seeing in a mitochondrial disease patient? Well, first of all, they have a very, very difficult time achieving 70% full capacity in that force transducer. So there's this myoclonic epileptic phenomenon, and there's a lot of shaking and twitching of the muscle. And as you might expect, very, very little deoxygenation, very little increase in deoxyhemoglobin, and very little deoxygenation of the oxyhemoglobin. And this just shows it a little bit more sort of all side by side, also showing that we have a diabetic subject here that shows a similar response. So let me kind of move to the last part, which is in between. And so literally in between, there is a lot of work going on in our community in breast cancer. And Jack, again, inspired much of that work. And as you heard earlier, he also inspired work in the MR community done by Mike Garwood, looking at early metabolites, choline response as a predictor of chemotherapeutic response. So what's going on here? Penn has long been a pioneer in this area with Brit and Arjun. This is one of the instruments that Arjun has built here, and he's very active in working with Mitch Schnall in integrating this technology with MRI. So there are many different opportunities. Early on, Jack became interested in some of the mathematical problems that are posed with respect to understanding boundary conditions, and he worked very closely with John on that. Hopefully you'll show us a few of those boundary condition solutions, John. This was in the early 90s. So this is a contemporary instrument that's in Arjun's lab. Here is our instrument that's similar in what it measures, but it's done at the bedside without compression in a reclining geometry. And essentially what we do is a point scan, location by location, and then can reconstruct maps of absorption and scattering in every region where we place the probe. And we can also construct contrast maps of oxyhemoglobin, deoxyhemoglobin, and so forth. And one of the interesting contrast indices that we find is a composite of water, deoxyhemoglobin, and lipid. This happens to be a very, very high contrast index for tumors that integrates both metabolism and structure to some extent. So we've explored this, the spectroscopy of breast cancers, and in this 58-patient study led by my colleague Albert Cerruti at UCI, we see dramatic differences in absorption spectra between malignant tumors and normal breast tissue. Now, one of the key questions is differential diagnosis. What about malignant tumors versus benign tumors? And I was just talking to Arjun, and he said that he just recently submitted a paper where they are looking specifically at malignant versus benign tumors and have had lots of some excellent results with that. So this is some of the reason why is that tumors do stratify with age. So the spectra are changing according to the physiologic status. Those are just malignant tumors. And looking at malignant versus benign, as you might expect, there's still spectral differences. One of the things that we've done to try to identify what these differences are between malignant tumors and benign tumors is look at how well those spectra fit to the basis spectra, the pure components. And in a technique that we call double differential spectroscopy, working with my colleague Enrico Graton, what we've seen is that there are key regions where there are significant differences between the basis pure component spectra and the spectra that we measure for malignant tumors versus benign. So in this hemoglobin region, we see significant differences. In this lipid region, we see very significant differences. And in this water region. And the implication of this is that the microenvironment, the milieu of those molecules, is different in malignant tumors versus normal, and this is without imaging. So this is simply looking at spectral separation. So the last example of where this is going is to treat locally advanced disease. And the question is, can we use optical imaging to predict pathologic response? Here is an MR image showing a volumetric change in this tumor after 4 cycles of an anthracycline and Cytoxan, AC therapy. This is given in many tumors, stage 2 to 4 tumors, and estimated to be a strategy that could impact as much as 20 or even higher, 25% of all breast cancers. So one of the key questions is, and increasingly oncologists are giving multi-stage therapy, is how do we figure out how patients are responding to this therapy? And is the first stage the right therapy? Should I switch stages to a new therapy? Is the cocktail an appropriate one? Is it causing an effect on the tumor, or is it causing just a negative effect on the patient? And about a third of patients are unresponsive. So I thought I would take you through just one patient to help you see sort of the power of this approach. So here is our baseline measurement of deoxyhemoglobin in a patient who's about to receive neoadjuvant chemotherapy. All these plots will be for neoadjuvant, for deoxyhemoglobin. And you see on day 3, after the first infusion, already a significant reduction in deoxyhemoglobin. And this is really quite dramatic. And it's very, very difficult to make these measurements in patients using MR, dynamic contrast MR, and to do them on a daily basis. No one's really ever looked on a daily basis because there hasn't been technology accessible at the bedside to assess the physiologic dynamics following chemotherapy. So here is day 4, and you already see a change between day 3 and day 4 in this patient. And suddenly, look at day 5. So there is a reappearance of these elevated levels of deoxyhemoglobin, which is not a good sign in terms of helping understand what's going on, or it's not a good sign for indicating that the tumor is responding. If deoxyhemoglobin is increasing, then cellular metabolism is high. We'd like to see deoxyhemoglobin drop. And here we are 1 week, and we see that deoxyhemoglobin has actually returned to and in fact exceeded the original baseline levels. So here we're moving along, and this is the first time anyone's really looked at the effect of Avastin using optical methods. So this patient received a second round of chemotherapy with Avastin and a few other agents. And we were very interested in comparing, in particular, the Avastin response optically to that of dynamic contrast enhanced MRI. So this is a week before Avastin. This is a week after the first Avastin. Overall, this is day 35. And we see that there's been a reduction in the deoxyhemoglobin level. That seems to be persisting. But maybe around day 12 after Avastin, day 39 overall, it appears to be coming back. And this is the overall long-term monitoring for that patient. And you can see amazing, really very interesting fluctuations which mirror what people see in animal models. In particular, Mark Dewhurst's group at Duke has explored this on a very, very dynamic basis and has seen physiologic fluctuations. So this calls into question, when is the best time to make a clinical decision? Is it here? Or is it here? At a high point, at a low point, and when should one make a measurement? So far we've done 20 subjects in this dual stage therapy with an Avastin agent in that cocktail, and have seen that if we look at the responses at the end of the first stage of therapy, after all the AC is complete, those responses are an excellent predictor of whether a patient ultimately, pathologically, will be a non-responder, a partial responder, or a complete responder. Over time, this separation decreases, but there's still very good separation by the end of chemotherapy. However, it kind of suggests that if we can pick a good midpoint and look at the response at that midpoint, we might be able to predict pathologic response, which ultimately is linked to survival. Now, these are results where we just ask the question, what happens if we make measurements at 6 days and we just forget about the rest of the chemotherapy? We just look at 6-day response. And this shows you in just 11 patients that we can achieve perfect prediction for a stratification of only responder versus non-responder. So this is a binary stratification, just by the measurements at day 6, based on all of these quantitative parameters. And how do we compare this to MR? Well, here is a patient who is a partial responder. The MR, you can't see it very well because there's too much light on. The MR does not indicate that there's any tumor. This is the tumor before. There's nothing in the dynamic contrast enhanced MR image. But in the optical image, there's still residual tumor. And we think this is a consequence of the way that Avastin disrupts the vasculature and impacts the delivery of the contrast agent to the tumor. So what about the future? Where is optical imaging going? And certainly Jack has inspired many of these concepts. And Penn has been a central player in all of these technologies, ideas, models, and computation that have emerged in our field. So first, in therapeutic monitoring, we think that we can have portable, individualized therapies that can help us understand therapeutic mechanisms. So one can potentially look at the response of these parameters and correlate that with mechanisms of action of drug, and allow us ultimately to do optimal drug dosing and perhaps even new agent testing as they become available in individuals. And for detection and diagnosis, it looks like optics will potentially impact this issue of how to screen individuals who are at high risk, particularly people who have high mammographic density. So perhaps it would be a tool to screen in those populations and maybe inform and enhance the use of dynamic contrast enhanced MRI. Perhaps one does not want to have that all the time, but an optical probe, an optical measurement could be the indicator that one should be, that an MRI should be obtained. So finally, I'd like to acknowledge all of my colleagues at UCI and at UCSF who've contributed to this work, and kind of conclude, really going full circle. Jack not only embodied sort of brains and muscle, but he studied them, and he studied with all the spectroscopies, which is really amazing to me, and he sure had a lot in between. So thank you very much.
Thanks, Bruce. The last speaker of the afternoon is going to be John Shotland. Most of you know John. John was a graduate student, as my young techs would say back in the day, with many of the others you've seen, Lizanne and Jim and Perry and others. Although John had the distinction of writing probably the shortest thesis I've ever seen, and probably doing the fewest experiments, right? 0, that's right. He got away quick. But anyway, John was very involved in developing a lot of the basic mathematical formalism and theoretical underpinnings of photon diffusion. When he finished his degree, took about a circuitous path, which included a couple of faculty positions, medical school, a couple things like that, and ultimately came back and is a faculty member in the bioengineering department, and so we're happy to have him back. And he's going to talk to us a little bit about some of the work that he injected in those areas. So, John?
Can you all hear me with this? Is this, maybe I need to turn it on. No, it seems to be. Okay, great. So I'm one of Jack's many academic children. And, you know, in sort of trying to think a little bit about what I wanted to say today, of course, there's almost a trivial statement that it's impossible to sort of summarize in one word what Jack meant to many of us. And in fact, I don't believe there is such a word in English, but I believe maybe there is such a word in German. And the word is Dr. Vater, or Dr. Father. I think that they don't make them like this. They broke the mold. And he is absolutely an inspiration for those of us who now have students and postdocs of our own. So what I'd like to do, first of all, is tell you about how I met Jack. And what happened was that when I was a first-year graduate student, I went to a talk in the physics department in DRL, and the talk was called The Physics of Proteins. And the guy who was going to give the talk, I'd never heard of before, and he sort of ambled in. And in DRL, the lecture halls often have large experimental tables for demonstrations, and he sort of jumped up on the table and crossed his legs like this and started to talk. So in those days, you know, there was no PowerPoint, but there were projector slides and overheads and even blackboards, But Jack had no need for any of that sort of technology because he was Jack, all right? And so he began to talk about proteins. But he began to talk about proteins in the way that only a physicist could, despite the fact, I think, that everything he knew about proteins he had learned from various sorts of people like biochemists, Britt and Mildred and so on. And the talk was mesmerizing. He, at the end of the talk, really made you feel as if you knew what it was like to live inside of a protein. You could feel the vibrations at the femtosecond scale. You knew about the rotations on other scales. You knew about electron transport and so on. And it was a remarkable performance. So I sort of bring up this story for two reasons. First of all, I think it illustrates that Jack's great charm. Right? The talk was utterly charming. And second of all, I think that it illustrates the fact that Jack had tremendous physical understanding. In fact, I would say that his physical understanding and intuition was second to none. It was remarkable. So I didn't see Jack after that for a long time, maybe about a year, when I went over to meet Britt, who was supposed to be my thesis advisor. And Britt took me down the hall and immediately introduced me to Jack, and then I had the canonical experience. So Jack said to me, you know, what do you like? And I said, well, I like physics and math. And he said, physics is okay, but, you know, mathematicians are the most useless people in the world. So maybe I agree with that to some extent. But what I would say as a caveat is that perhaps mathematicians are useless, but math isn't. Okay. Anyway, despite Jack's sort of usual iconoclastic ideas about math, he was in fact delighted by numbers. And he was almost a numerologist in some ways, which can be a dangerous profession. So at one point, I think when I was just about finished with medical school, he told me that, you know, there was a house across the street from him that was for sale, and I had to buy it because, you see, its address was 346, and he lived at 345. And those of you who know that 345 is a Pythagorean triple, and this absolutely made Jack very, very happy. So we started to talk about, well, how many Pythagorean triples are there? Of course, I'd never thought about that before. So Jack said, well, you know, of course, there are infinitely many of them. You can take any Pythagorean triple and rescale it, and you'll get another one. But let's forget about all those. Let's think about the non-trivial ones, the sort of irreducible ones. How many are those? So I started to calculate what that was, and we found out that they're exponentially rare. Now, there are similar questions that come up in number theory where you might ask what is the distribution of primes and that sort of thing, and those are much harder questions. But Jack didn't believe me because I had only done a calculation, so instead what he did was a little computer simulation, and he searched for Pythagorean triples, and he checked that the density that we calculated was in fact correct. So that's Jack. He was distracted by everything and brilliant at once. So, what I want to say, I guess, is that Jack, nevertheless, despite his feelings about mathematics, seems to have had a tremendous influence on those of us who are sort of mathematical scientists. And this is true particularly in the area of inverse problems. So let me tell you what inverse problems are. So inverse problems are a branch of applied mathematics that's very, very closely connected to imaging. So it's like the game of Jeopardy, right? So you tell me the answer, and I have to guess the question, right? So these are problems that are inside out, so to speak. Okay. So let me tell you that perhaps the most challenging inverse problems, at least that I'm aware of, that come up in imaging, are those which are important in optical tomography. And I had a wonderful speaker, I couldn't have done better, who introduced all of the important ideas ahead of me. But let me just say a few things about this. So Jack, of course, was among the first to realize that there were even things to think about in this area. And one of the things that I don't believe that he ever knew, but he might even have been embarrassed if he knew, is that now there are many, many meetings about inverse problems in the applied math community, and a mainstay of many of the discussions at those meetings has to do with the inverse problem of optical tomography. Okay, so let me try to move to the blackboard. So let's begin with a very, very simple experiment. Okay, it's an experiment that only somebody who never did one could ever describe. So suppose we have a glass of water or something like that, a medium which is completely transparent, and inside of it, it has some inhomogeneities that you're interested in. Okay? So little marbles or something like this. And now suppose we illuminate this on one side, and we measure the light which comes out on the other. So remember, it's completely transparent. So what do you see? What you see is just, if you measure the intensity over here, you see just a little projection. Right? So wherever there's something which can absorb the light, you lose something, and something like that will happen. Okay? So it's just like X-rays, right? So everything you know about X-rays, if you're a radiologist, you should think of light as behaving this way when there's no scattering, when the medium is transparent. Okay. Now, suppose we add a little bit of milk to this little container here. What happens is that it becomes immediately cloudy. And in fact, even a little bit of milk makes it impossible to see through. It obscures what's inside. Okay. So what's inside is really a proxy, as Bruce told you, for things like tumors in a breast, right? So these might be the tumors. But instead of measuring a nice projection image, what happens is that if the light is coherent, for example, that you get what's called a speckle pattern. Okay? And that speckle pattern doesn't look anything at all like what's inside. Okay. But Jack wanted to know what was inside. And so he had a very important idea. He said, well, and Bruce alluded to this, he said, suppose we do an experiment where we have a very fast laser which makes a pulse, and we measure the light that comes out over here using some sort of time-resolved detection. Okay, so it's like a fast camera. So if I plot, the arrival time of photons, okay, so this is the number of photons which are detected, and this is the time after the pulse. Well, initially, there won't be any photons, but then the number of photons grows up, and it finally falls off. Okay. So what's going on around here? Well, so if we think about a source here and a detector over here, people can see this. Can you see this? Then a photon which comes through in very short order has not scattered too much, and so it takes a path which is nearly straight, or sometimes these photons are called ballistic. And Jack's idea was that if you could somehow select the earliest arriving photons and throw away the rest, that you could make an image of what's inside. Okay. And so he suggested that, and he and Britt wrote that paper that Bruce had in 1988. Now, so at some point, we started to talk about this, and he said, well, we should just be able to do tomography now, because we should be able to do lots of such experiments from many different sources and detectors and combine them all in some way and make an image of what's inside, a quantitative image of, let's say, the optical properties of the medium. And my reaction to that at the time was, that can't possibly work. And the reason for this is because it turns out that the transmission, the fraction of photons, ballistic photons, which go through a medium of this size, let's call it L, falls off exponentially on a scale which is called L star. It's about 1 millimeter in tissue. So more than a centimeter or so of tissue results in nearly unmeasurably small signals. Okay? And this was known to experimentalists, of course. But Jack said, no, no, no. What about all of the other light that comes out at longer times? He was an iconoclast, of course. And so what about all these photons that come out near the peak? So they take long and winding paths before they get from the source to the detector. And there it turns out there are a lot of them. Their intensity, let's call this the ballistic ones, and call these the diffuse ones. Their intensity falls off roughly algebraically with the size of the system. So there's a lot of light, but as I mentioned earlier, there's a significant question about whether there was any information in that light. Again, I told Jack I didn't think so. But Jack knew better. So, meanwhile, while I was trying to prove to Jack using mathematics, because after all, mathematics is something that you use if you're not as smart as Jack, this possibly couldn't work. He and Mark Elliott started to do Monte Carlo simulations to show that somehow it could work. So they launched a lot of photons from a source, and they measured where they wound up in some detector, and they made a picture in which they plotted the density of such photons at every point in space. So it's concentrated along the line connecting the source and detector, and it gets more and more fuzzy the further away you go. And Britt called those things bananas. Okay. So meanwhile, as I say, I was trying to show that this couldn't possibly work. And I went back to thinking about regular tomography when there's no scattering. So one of the things that you find out is that if there is no scattering, that the intensity of photons falls off exponentially with absorption. And so if they're little absorbers, you can then pick a ray, let's say, which passes through one of them. And the loss of intensity along that ray obeys the following relation. It says that it's simply proportional to the integral of the absorption along the ray. Okay? So there's this exponential loss. So what should we do in the case when the paths aren't straight, but they're much more like Brownian paths, right? They're much more like these diffuse paths that Bruce was talking about. So the idea was, in the case of diffuse light, well, you shouldn't integrate along rays. What you should do is integrate the absorption along a path and then weight that integral by all the possible paths that a photon could take, right? So there are all of these possible paths. They're sometimes called Feynman paths. And so you need to integrate with respect to all of those paths to sum them up. Okay, so that's a formula. It tells you in a very, very scattering medium how to compute the result of an experiment. You say, if I have a source somewhere and a detector somewhere and I know it's in the medium, I can predict what the transmission of light through the medium is. Okay. So when I wrote this down and did a little bit of calculation, what I found were bananas. How did Jack know that? That was a big question for me. How did he know? He just did. So we were halfway there at that point. So we knew how to predict the result of an experiment which meant that in principle it should be possible to solve the inverse problem. So that says, how do we find alpha if we measure the transmission from many, many measurements? So, well, that can be converted into something called an integral equation, and the integral equation can be solved. And we tried the simplest thing you possibly could, which involved eventually putting it on the computer. So along with not knowing how to do experiments, in those days I also didn't know how to program a computer, but Jack did. So we programmed the thing up, or he programmed the thing up, and we picked some distribution of absorbers inside of a little two-dimensional phantom, which only existed in the computer, of course, and we solved that equation for the absorption. And I fully expected that when we tried to invert the equation and recover the absorption, that we wouldn't see anything, right? That there wasn't, in other words, that there wasn't enough information in the measurements. But Jack was right. He knew all along. And when he pushed the button the first time, we had an image. And it was one of those moments I'll never forget. So how did he know that? I still think about that. Anyway, that launched me, like John Detra, sort of one good idea from Jack, on a career in which I've thought for a long, long time now, maybe 15 years or more, about why it is that it's possible to invert such a thing, why it is possible to get information. Because our numerical experiment only demonstrated feasibility. It didn't give us an answer which was mathematically satisfying. And so, sort of, I'll spare you the details, but one can say that there's now a very rich, interesting, at least to us, who do these kinds of things, mathematical theory of why this can be done. And it has spawned developments in all sorts of other areas of inverse problems and in optics beyond the initial applications that we had thought about. So Bruce mentioned that the diffuse waves are a little bit different than standard electromagnetic waves. They're sort of like the modulation envelope of his little oscillator. And so they don't propagate the way standard waves propagate. They fall off exponentially with distance into the medium, and the highest frequencies die fastest. Okay, so there are other waves, real waves, that behave that way in optics, and they're called evanescent waves. And they provide information on scales which are small compared to the wavelength. And so there is now quite a bit of activity in the area of trying to recover information about scattering objects on nanometer scales. And the mathematics is almost the same as the mathematics that comes up in optical tomography. They're very close cousins. And the story goes on, but we have food to eat. So I'll stop here. Thanks very much.
Well, I really want to take this opportunity to thank everybody who's here today and all of the speakers who I think did an absolutely superb job in taking us through a little bit of Jack as a person and Jack's science and the impact that his science had. A couple of months ago when we sat around and tried to think of what we were going to do to mark Jack's passing, as many of you know, he left explicit instructions. There was to be no traditional service, funeral, anything like that. And although we all felt the need to do something, we also felt the need to honor his wishes and to honor the intent. But we thought Jack would like science. He always loved science, so we should try to remember his science. And we organized this symposium, which I think has come off better than we could have ever hoped. And I think if Jack were here, I think he would have enjoyed it. I think he would have enjoyed the science. But I think what he would have enjoyed more, and I think you heard it in a theme that permeated almost everybody's talk, I think he would have enjoyed more seeing the people that he touched and seeing what they did and what their science was, because that's what Jack really stood for. Jack loved to teach, as you heard, and I think that he got his greatest joy. You know, when someone's thinking and they're having that close to eureka moment, you can look in their eyes and you can see the wheels turning. And I think Jack's greatest joy was looking into people's eyes and seeing those wheels. He loved, whether it was a student, whether it was a colleague, whether it was someone who was senior to him, or whether it was a child. I used to bring my children to the lab, and they loved Jack. Jack would give them little problems to do, and he would just watch them and just marvel as they would try to solve the problem. He just loved teaching people how to think and challenging them at whatever level they should be challenged, and pushing them to respond. Now, it wasn't all good, because the one thing Jack didn't tolerate were people who didn't want to think. And if you sat in and around Jack long enough, you got to see a number of those interactions where people wanted to come down. They wanted an answer. They didn't want to think. He never gave an answer. Okay? All he gave you was the opportunity to figure out the answer for yourself. And I think that, at least to many of us, was a great gift, giving us that ability to ask questions and figure out answers on our own. So we're going to have a reception following the symposium. At the reception, there's going to be a podium set up for people to tell their favorite Jack stories. Those of us who had the opportunity to take this podium, and I'll take an opportunity in a minute, get to have had that opportunity, but I think everybody, I'm sure, has great stories that they may want to share, and everybody's going to have that opportunity to share them. I think one thing that was alluded to in a number of the discussions that we heard, were Friday nights at the JF. And I don't know how many of you, but I suspect most of you, or a lot of you, had the opportunity to spend a Friday night. And what typically would happen is sometime in kind of mid-afternoon, a number of the students would go bumming dollars out for people so we could grab enough money to go out and buy beer. Because we were living on that, $421 a month, Jim? Is that what it was? So we had to go bum dollars to get beer. By the way, I thought I was rich at that time. So we'd go around the labs, and not just our labs. We'd go up to George Reed's lab and Tony Scarpa's lab. He's got this important position running CSR, but he drank beer with us too. And bummed our dollars, and then we went out and walked a couple of blocks, and like a bunch of dummies, carried 4 cases of beer back by hand a couple of blocks from the closest beer distributor. And then we all sat around and just talked, and we talked about just about everything, and Jack made everything into a very serious intellectual conversation, whether you were talking about unusual pronunciation of scientific authors' names that made them sound like things we shouldn't be talking about, or whether it was the interesting spiral shape of the porcine phallus. It always boiled down to something that was critically important, that was going to change the world. But it really, I think, created that easy environment that allowed us all to have fun together, work hard together, and really enjoy each other. So I'll conclude the seminar and invite everybody to join us back where we had lunch. There'll be a reception and an opportunity to talk and tell our favorite stories. Thank you, everybody.